Good afternoon, children. Let's start very quickly. Today I'll take a test also. Today is Saturday. I was supposed to take a test yesterday. I did not take about working of hormones. That part I'll be taking a test today. Later in the class, I'll be taking a test. Remember, what is the lifespan of an RBC?
120 days.
20 days.
120 days.
Name the cells in the liver which help in the disintegration of RBCs.
Copper cells.
Yes. A healthy individual has how many in 100 ml of blood?
12 to 16 grams.
15 grams.
In per cubic millimeter of blood in men?
100 million.
No.
5 to 5 million.
Yes, 5 to 5.5 millions. So this is all about RBCs. Very quickly, we'll be doing about WBCs, and then I'll take a short test today based on the function of hormones. Leukocytes, they're known to be the most active and motile constituents of blood as well as lymph. What is motile?
Movable.
Yes. And why are they white? Why are they not red?
Because they don't have hemoglobin pigment.
Yes. That red color pigment is not there. Which was that red color pigment?
Hemoglobin.
That is the reason they are colorless. RBCs were red because of the presence of pigment, so they are colorless. Let's come to the next one. These are nucleated and are generally short-lived cells. We'll keep comparing. What do you mean by nucleated?
Have a nucleus. Which have a nucleus.
They have a nucleus. They're short-lived cells. The number of WBCs are relatively lesser in number. About how much per millimeter cube of blood? Tell me quickly.
6,000 to 8,000. 6,000 to 8,000 millimeter cube of blood.
And the average was what? When we talked about RBCs, what was the average? Keep comparing side by side.
5 million to 5.5 million.
Yes. What was the lifespan of WBCs? What is
120
120
days.
days.
because this is 13 to 20 days only. 13 to 20. That is why we call them as short-lived cells. So what is the lifespan of an average lifespan of a WBC?
13 to 20 days.
13 to 20 days. Now, they move in an amoeboid fashion. What do you mean by amoeboid fashion?
Like amoeba.
Like amoeba. So they move in such a fashion they can squeeze through capillary walls and move to the site of action. So they move in this amoeboid fashion. So they can easily move through the capillary walls and then they reach the site of action. This phenomenon is called as diapedicus. This is very important. Okay. We should know what is diapedicus. Let's do what is diapedicus. You read for a second and then let's talk about it. Vedvida?
Yes.
Okay. See, when your blood cells pass through the walls of the capillaries, okay, sometimes it leads to inflammation also. This leads to inflammation at times. Then this process is what we call movement of the WBCs in an amoeboid fashion through the walls of the capillary, mostly leading to inflammations. This process is diapedicus. diapedices. So when I say diapedices, you should remember three things about diapedices. What is moving in diapedices? Which blood cells?
WBCs.
WBCs.
Even even RBCs move, but it is mainly the WBCs. So here it is the movement of the white blood cells in what? They move through which walls?
Capillary wall.
Capillary walls.
Yes, yes. And generally lead to inflammation. If you remember, I told you whenever there is an injury, the body prepares itself with the help of inflammation process. We had discussed this earlier. So this is how diapedices is going to help you. Whenever there is a tissue which gets injured, so diapedices is basically a reaction of the body for that. Reaction of the body for against injuries. This, it will immediately reach where? They will move and they will reach the site of action, the damaged tissue. This process is called as diapedices. The white blood corpuscles are categorized into two main categories. The first is granulocytes. Okay, what does the word give you an idea about granulocytes?
They are granular.
Which have granules in cytoplasm.
Yes, you get an idea. They contain granules in the cytoplasm. That is why we call them as granulocytes. They have regularly lobed nucleus. What is a lobed nucleus? We did lobes in some structure, if you remember, when we did glands.
Pituitary gland.
Yes. So here, what structure is being talked about being lobed?
Granulocyte.
Yes. In
Granular.
granulocytes, it is the nucleus which is lobed. If you see this word, what does it say? Lobed nucleus. What is the lobed nucleus?
Which has lobes.
See, beta, where the genetic material, that is the DNA. Normally what happens is we have one nucleus and all the genetic material is inside that nucleus. But sometimes in some cells, the nucleus, what we have is that the genetic material is packaged in several spheres instead of a big sphere. Normally, we have a big sphere, that is your nucleus. Inside that, we have all the DNA. But in some cells, what happens is the DNA is segregated in multiple spheres. It is not just one sphere. Such kind of a nucleus is known as which nucleus? It is known as a lobed nucleus, or it is also known as a multi-lobed nucleus. So what are the main identifying features of granulocytes? They contain granules and they have which kind of a nucleus?
Multi-lobed nucleus.
Yes, beta, the nucleus is which kind of a nucleus? It's a lobed nucleus. Again, the granulocytes are further divided into three main types. The first are neutrophils. The most abundant cells here are neutrophils of the total WBCs. Total WBC 60 to 65% is neutrophils. They stain equally well with acidic as well as basic dyes because they're neutral in nature. What is their nature?
Neutral.
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 So they can stain very well with both the dyes. These are phagocytic cells that destroy foreign organisms entering the body. What is the process known as? When they engulf any foreign organism which enters the body?
Phagocyte.
Phagocytosis. You remember this term? Yes, it is phagocytosis. So, which are the main ones when we talk about WBCs? Which are the main cells?
Neutrophil.
Neutrophil. How many percent?
60 to 65.
Yes. Let's move on to eosinophils. They have a bilobed nucleus. Bilobed means here how many segregations? The DNA would be segregated into how many spheres?
Two.
Two.
Very good. So multilobed, bilobed, now you know. Eosinophils are bilobed. They are stained bright red in color with acetic dye due to the presence of numerous coarse granules in it, such as eosin. So which is the stain generally which we use to see eosinophils? Eosin. That is why we call them as eosinophils. If you want to see eosinophils, very easily you stain with which dye? Eosin. So if you'll stain with eosin, you can see them bright red in color. This is an acidic dye. Eosin is a acidic dye. So you can see your eosinophils very clearly, but they are only 2 to 3% of the total WBCs. They resist infections and are also associated with all allergic reactions. They help in dissolving the blood clots. They help to destroy the toxic substance present in the body. And during allergic conditions, the number of eosinophils increases in the body. So basically, they're helping you to fight against diseases if you see allergic reactions basically, and in dissolving the blood clots. How many percent of the total WBCs are eosinophils?
2 to 3.
2 to 3%. So we have done how many kinds of granulocytes till now?
2.
2.
Which was the first one, which was 60 to 65%?
Neutrophils. Neutrophils.
The second ones we did was?
Eosinophils.
Yes. Moving on to the third and the last category of this, that is basophils. This is the last category. They contain fewer coarse granules than the eosinophils and can be stained with basic dyes. If you remember, we had stained eosinophils with which dye?
Acidic dye.
Acidic dye. What was the name of the dye?
Eosin.
Eosin.
But basophils, the name itself tells you base. Means you can stain them with which dyes? Basic dyes. This is how we remember basophils. So you can stain them with the basic dye. Which is the basic dye we generally use? It is methylene blue. There we had used which stain bidder for eosinophils?
Eosin.
Eosin.
Eosin is which kind of a stain? Acidic or basic?
Acidic.
Acidic.
And for basophils, we use a basic dye, that is methylene blue. They are found least abundant, 0.5 to 1% among WBCs. They secrete histamine, serotonin, heparin, etc., and are involved in inflammatory reactions. When we talk about the dyes, if you see the first one, they stain equally well with acidic as well as basic dyes. Means you can take any one dye, either take acidic or take basic. Now, very quickly, tell me all the three kinds of granulocytes. Which was the first one?
Neutrophil.
Neutrophils.
Neutrophils. How many percent?
60 to 65%.
60 to 65%.
Which kind of a dye should we use?
Acidic.
Acidic or basic?
Neutral.
Both.
We can use any. Why we can use any? There is no limited choice, because what is their nature?
Neutral.
Neutral.
They are neutral. So you can use any dye. You can use an acidic dye like
Eosin.
Eosin.
also use a basic dye like.
Methylene.
Blue.
Very good. So we have done about the first category, that is neutrophils. And what is the main nature they have? They destroy what?
Phagocytic.
Yes. They perform the function of phagocytosis. That means they destroy the harmful organisms. Let's move on to the second type of cells, second type of granulocytes. What are they?
Eosinophils.
Yes. Eosinophils. How many percent?
2 to 3%.
2 to 3%.
Yes. And you can stain them bright red with the help of a dye. What is the name of the dye?
Eosin.
Eosin.
And it is because of eosin that we simply call them as eosinophils. What is the nature of this dye?
Acidic.
Acidic.
Yes, it is acidic, and they're very less. We just did just 2 to 3%. And basically, they help you to fight against all allergic reactions. Now, let's move on to the third category of cells. What is the third category of cells?
Basophils.
Basophils. Which one has more granules? Basophils or eosinophils?
Eosinophils.
Eosinophils.
Yes, eosinophils have more and basophils are less. You can stain them with which dyes?
Methyl blue. Methylene blue.
What kind of a dye is it?
Basic dye.
Basic in nature.
Basic. And if you see, when we talk about abundance, then they're very, very less. Only how much percent?
0.5 to 1%.
1.
Yes. So we are done with granulocytes. Everybody remembers the 3 types of granulocytes. We are talking about WBCs. WBCs again into 2 categories, granulocytes and agranulocytes. Granulocytes, we are done, neutrophils, eosinophils, and basophils. Now we move on to the second category, that is agranulocytes. They lack granules. They don't have granules, beta. Okay. They do not have granules. So we simply know that they lack granules in the cytoplasm. They have rounded or oval nucleus. Here you are not finding any lobed nucleus, multilobe, bilobe, nothing you can see over here. As of now, if you see, agranulocytes are also subdivided into many types. What are the types? Lymphocytes and monocytes. What are the 2 categories of agranulocytes?
Lymphocytes and monocytes.
Lymphocytes too. Lymphocytes and monocytes. Lymphocytes, you know, they are smaller in size. They have a rounded nucleus. Lymphocytes are further 2 types, that is B cells and T cells. Both of these cells are responsible for immune responses of the body. So when we make a flow chart, we'll write agranulocytes into 2 types, lymphocytes and monocytes. Lymphocytes again of 2 types, B cells and T cells. And what is the main function of B cells and T cells?
Immune
So again,
responses.
Okay, immune
Antigen.
system of the body. I'll just be explaining this part again to you. And the second are monocytes. These are the largest of all types of WBCs, but are fewer in number. If you talk about the size, see this one. This is the biggest one. This is a monocyte. This is the largest in size, but then when we talk about number, they are few. Mature monocytes are known as macrophages. Okay, when a monocyte becomes mature, we call it as a macrophage. Macro means big, and phage, the cell which can cause phagocytosis. They help to kill foreign particles, and they're big in size, so that is why we call them as macrophages. These are phagocytic in nature. So let's go back very quickly. Agranulocytes, I'll give you 1 minute to read. Also see the figure, all of you, these kind of cells that we have. RBCs, platelets I have not done with you, eosinophils, basophils, neutrophils, monocytes, and T lymphocytes and P lymphocytes. Very quickly, I'll give you 1 minute read. Let's discuss it once again. Do you know the difference between B cells and T cells? The similarity is that both of them are responsible for immune responses. Any idea about what is the difference between B cells and T cells, children? Anyone? No idea? See, beta, the T cells can recognize only viral antigens. Okay? The T cells will be able to identify only viral antigens, and the B cells can recognize both antigens of bacteria and virus. And similarity, of course, you know, we just told you they are responsible for immunity. Now tell me the difference between B cells and T cells. T cells can only recognize which antigens?
Viral antigen.
Viral antigens. And what about the B cells?
It recognizes both viral and bacterial.
Yes, for B for bacteria, you can remember it like this. They can even identify the bacterial antigens. So they can fight against, they give you immunity against both bacterial and viral. Whereas T cells, they will just be able to identify which antigens?
Viral.
Viral antigens. So agranulocytes, I don't think I missed out any word which we need to discuss. They lack granules, they have round nucleus. They're divided into 2 types, lymphocytes, monocytes. They are smaller in size. If we talk about the two differences, we compare them. The lymphos are small, monos are larger. Lymphocytes are again of two types, B cells and T cells. The similarity between both of them is that they're responsible for immune responses. And there is, of course, a difference also. We just discussed the T cells can just identify the viral antigens, whereas B cells can identify both bacterial and viral. Monocytes are the largest of all types. See this big one. When the mature monocyte is there, it is also called as what? What is it called as?
Macrophages.
Macrophages. Because the word macro tells you it's very big in size. And phages because it is phagocytic in nature. We have already done about phagocytic. Third one is your blood cells, blood platelets. Let's finish this part today. Thrombocytes. These are cell segments produced from megakaryocytes. What are these megakaryocytes? Special cells found in the red bone marrow. This is a new word for you probably. Special cells which are found in the red bone marrow, they are known as megakaryocytes. Okay, speak out this word, children.
Megakaryocytes.
Where will you find megakaryocytes?
In the red bone marrow.
What are they responsible for?
Clotting.
See,
Blood
beta,
clotting.
I'll tell you why it is called mega, because it is a very large cell. Okay, it's a bone marrow cell, of course, red bone marrow cell, but it's a very large cell. And this also has a lobed nucleus. Basically, this helps you in production of platelets, and platelets are responsible for blood clotting. So it is going to help you where? It is going to help you in blood clotting indirectly. How? It is these megakaryocytes which will be producing what? They will produce the platelets. Okay. And now we are coming to this. What is it? That they're necessary and platelets are responsible for blood clotting. So let's very quickly do what is the other name for blood platelets?
Megakaryocytes.
Yes, very good. Thrombocytes. Thrombocytes.
Thrombocyte.
Thrombocytes are produced from special cells found in the bone marrow. What are these cells called as?
Megakaryocytes.
Megakaryocytes.
We call them as megakaryocytes because mega means big and karyo means nucleus. They have a large nucleus. That is why we call them as megakaryocytes, because they have a large nucleus. We call them as megakaryocytes. They are oval-shaped, disc-like cells found only in the mammal blood. They are devoid of nuclei. They don't have a nucleus. The platelets contain mitochondria, Golgi bodies, and some other structures such as granules, tubules, filaments of actin and myosin. Basically, you need to remember about this, that they're devoid of nuclei, but they do contain some important cell organelles like mitochondria and Golgi bodies. Let's talk about the number. Blood normally contains about, yes, how many? Quickly. Count the zeros so that you remember yourself.
1,50,000 to 3,50,000.
Okay, so the number is less or it is quite good?
More.
Yes.
More.
I just told you that megakaryocytes, we have the formation of what? Thrombin, which leads to blood clotting. So if there's a reduction in the number of thrombocytes, an individual will suffer clotting disorders. Clotting disorders, which will lead to excess blood loss from the body if someone gets injured. I think I had discussed about the royal family thing that day, if you remember. It is something this only. The lifespan of platelets is only about 7 to 12 days. Did we discuss about hemophilia that day a bit? Blood clotting disorder?
Yes, ma'am.
Yes, ma'am.
The lifespan of platelets is only about 7 to 12 days. And the formation of thrombocytes is also called as thrombopoiesis. So what is thrombopoiesis? Formation of thrombocytes. It is also called as thrombopoiesis. Since we have done this, let's compare both of them, RBCs and WBCs. They're small in size, they're large in size. They're more numerous in number, okay? That is the number of RBCs per cubic millimeter of blood is average 5 million, but here it is only 8,000. They are non-nucleated, WBCs are nucleated. This is one more shape. Now we said amoeboid cells, that means they can change their shape. Like amoeba can change their shape, WBCs can also change their shape. RBCs contain hemoglobin, WBCs do not contain hemoglobin. RBCs cannot diffuse out through the capillary wall, WBCs can diffuse out through capillary walls. What was that called as? Let me see if you remember, anybody.
Diapedicil.
Yes, very good, very good. We had done this just now. Too good. RBCs help in the gaseous transport, WBCs help in killing bacteria and foreign particles. Which other foreign particle you think can they kill?
Bacteria and
Bacteria.
viruses.
Yes, bacteria and viruses. Which cells will help to identify bacteria?
B cells.
B lymphocytes.
Better. RBCs settle together to form Rolex, stack over each other due to abnormal shape. What is a stack?
Pile.
Yes, it's a pile. You take one book, keep another on it, another, keep keeping it in a uniform manner, then it's what we call as a stack. But if you keep it in a haphazard manner, it's not organized. Abnormal shape you get. You don't get a normal pillar shape. Then this is known as a Rolex. What is it called as a Rolex? So RBC is one over the other, one over the other. They of course stack over, but the shape is not normal. It is not organized. So we call it as Rolex. RBCs settle together to form Rolex. WBCs do not form a Rolex. The stacking does not take place. Stacking does not take place in WBCs. So very quickly, we've got the differences in the size, the number, the nucleus, the hemoglobin, the diffusing in the capillary walls, the functions, and the property of Rolex. What is Rolex? Stacking in
Stack
which
over each
manner?
other.
Yes, stacking in an abnormal manner. So Rolex property is so shown by RBCs or WBCs?
RBCs.
RBC.
So how many differences? I'll give you 1 minute. Please analyze this table, then I'll start discussing the differences with you. Start, beta. Start, all of you. See it nicely. We'll discuss the differences. See quickly. Count how many differences are there.
Six.
6 differences.
6 difference.
If I hide this, anybody can give me all the 6 differences? Anybody would like
Yes.
to try? You can take a minute extra. I'm absolutely okay. Let me see. I'll hide it and try and give me the differences. Raise your hands, whoever is confident, then I'll hide. Srishti and Mansi, 2 hands. Okay, fast. Let's start. First, let me hide Srishti. Two hands I saw. Let me see if there's somebody else also. Srishti and Mansi only as of now. I hope you win the challenge. Others, please keep listening carefully. If you find mistakes, you will get a point. Start. First, Srishti.
Ma'am, RBCs are smaller in size, whereas WBCs are larger in size. RBCs contain hemoglobin, whereas WBCs do not contain hemoglobin. RBCs do not have a nucleus, whereas WBCs have a nucleus. RBCs are biconcave and disc shaped, whereas WBCs are amoeboid by nature. RBCs cannot diffuse through capillary wall, whereas WBC can diffuse. RBC uh, RBCs form Rolex, whereas WBCs don't, and RBCs help in gaseous transport, whereas WBCs help in defensive mechanism. And
How many
RBCs
bits?
have
Continue.
5 million 5 million cubic millimeters of blood. Their number is 5 million cubic millimeter of blood, and WBCs is only 8,000 cubic millimeter average.
Did she miss out on something, children? I don't think she did. As far as I know, she spoke all of them. Okay, Mansi, your turn. Start, beta. If somebody else also would like to try, raise your hands. Yes, Mansi, start.
RBCs are smaller in size, whereas WBCs are larger in size. RBCs are 5 million per cubic meter in blood, and WBCs are 8,000 cubic meter in blood. And RBCs are non-nucleated and biconcave, whereas WBCs are nucleated and amoeboid cell. RBCs contain hemoglobin, and whereas WBC do not contain hemoglobin. RBCs can diffuse out through capillary wall. WBCs cannot diffuse out through capillary wall. RBCs form Rolex due to abnormal shape, and WBCs do not form Rolex. RBCs help in gaseous transport, and WBCs help in killing foreign particles.
She's given all. I don't think she's missed out on anything. Anybody else would like to try? No? Nobody wants to take up the challenge today?
Ma'am, she
Fine.
gave a wrong point.
Which one, beta?
Ma'am, she said, ma'am, WBC ma'am, cannot diffuse through capillary walls.
She said it opposite.
Yes, ma'am.
Okay, okay. While speaking, she must have got messed up, no? Either ka udhar she must have said. That's okay. How many of you, we have last 15 minutes left. How many of you are ready for that test which I said? Slow acting and quick acting hormones. Raise your hands. You have to simply explain on a paper how slow acting hormones work and how quick acting hormones work and post on the group. That is all you have to do. How many of you are confident? You have to draw what I had drawn, explain in your own words the difference in the slow acting and quick acting hormones. Now raise your hands. How many of you are ready? I was supposed to do it yesterday, I told you, but I did not take the test yesterday. Quickly raise your hands. Hurry up. Vedehi, Ariba, Srishti, Akash, Ananya. Hurry up. Akash, Drishti, Mansi, Soni, Uditi. What happened? You all are not ready. You have to simply take a paper, explain the working of slow acting and fast acting hormones and post it on the group. That is all you need to do. Okay, one more hand. Akash, Udrishti, Mansi and Soni. You people are not ready? Soni has already joined us. Akash, Drishti and Mansi left now. Beta, you will not be able to try also. Oh, very good. Excellent. So now all of us, even if the meeting gets over, I'll start the meeting again. You have to write your name on the paper. You have to write your name on the paper, and then what you need to do is you'll post on the group, and I'll post your marks today itself on the group only. So are we all ready?
Yes,
Are we ready? We have 15 minutes. So you can start your work. Even if the meeting gets over, no issue. I'll be starting another meeting. But you have to do this work. Take a paper. You have 15 minutes. When you finish, post on the group. This is a test. Please start last 15 minutes. Please start your test. Don't bother about the meeting getting over or something. I'll start it again. Start, children. A mechanism of quick acting hormones and slow acting hormones. You need to draw and explain in your own words. I don't want you to copy it from anywhere. That is not needed. Please start, beta. And then post on the groups only. I have the name of students who are the participants. Everybody needs to post.